This is your instructor, Skylar Huff. We've finally made it to the last chapter of the semester. Or in the event you've taken the course during the fall, this will not be the last chapter of the semester. As we begin, I'll go through these key concepts quite briefly. First things first, the flower is the site of sexual reproduction in angiosperms, and there are 4 worlds that make up that typical flower. They are as follows: sepals, petals, stamens, and carpels. Next, pollen grains are transported to stigmas by a variety of agents such as animals or wind. Up next is that double fertilization results in a plant embryo and an endosperm. The seed is that mature ovule, and the fruit is the mature ovary. Following a period of dormancy, a seed germinates or sprouts, and then the young seedling becomes anchored in the ground and begins to photosynthesize. And then thereafter we'll get to many vegetative organs, such as roots, stems, and leaves, are modified for asexual reproduction. And then lastly, sexual reproduction in flowers results in genetic variability in offspring, whereas asexual reproduction in the vegetative structures generally results in individuals that are genetically identical. Let us begin. So the flowering plants, or angiosperms, include an approximate 300,000 species and are the largest and most successful group of plants. You may have admired flowers for their fragrances as well as their appealing colors and varied shapes. The biological function of flowers is sexual reproduction. Their colors, shapes, and fragrances increase the likelihood that pollen grains, which produce sperm cells, will be carried from one plant to another. Sexual reproduction in plants includes meiosis and the fusion of reproductive cells, egg and sperm cells, collectively called gametes. The union of gametes, called fertilization, occurs within the flower's ovary. Sexual reproduction offers the advantage of new gene combinations not found in either parent that may make an individual plant better suited to its environment. These new combinations result from the crossing over and independent assortment of chromosomes that occur during meiosis, before the production of egg and sperm cells. Many flowering plants also reproduce asexually. Asexual reproduction often does not involve the formation of flowers, seeds, and roots. Instead, offspring generally form when a vegetative organ, such as a stem, root, or leaf, expands, grows, and then becomes separated from the rest of the plant. Often by the death of tissues, because of sexual reproduction requires only one parent and no meiosis or fusion of gametes occurs. The offspring of asexual reproduction are virtually genetically identical to one another and to the parent plant. So here we'll examine both of which, being sexual and asexual reproduction in flowering plants, including the floral adaptations that are important in pollination, seed and fruit structure, and dispersal, germination, and early growth, and several kinds of asexual reproduction will round out the chapter class. So with that, let us begin. So please put your mind back where we were with the alternation of generations, keeping in mind that all plants alternate between that haploid gametophyte generation, of course being multicellular, and that diploid sporophyte generation. Keep that in mind because the very same thing you learned then, you'll learn now. And do look for the figure that shows the way in which those generations alternate. In case you don't know what I'm referring to, if you take yourselves back to the 28th chapter, I say, to make sure you are pretty well prepared for the exam, please take yourselves back to the 28th chapter. And in chapter 28, where you first learned about the way in which generations alternate, I also mentioned during that lecture that you should know and be able to fill in the components for the sexual reproduction in angiosperms for your fall exam. If you don't know what I mean, in your textbook, please turn to page 595 and see figure 28.13. So those things that are numbered you should know, as well as those things that are not numbered, that provide us with the way in which those generations alternate in angiosperms. This is the life cycle for a flowering plants class, the key point. This is the way in which the flowering plant life cycle has double fertilization, in which one sperm cell unites with the egg, forming a zygote, and the other sperm cell unites with two polar nuclei, forming that triploid cell that gives rise to the endosperm. So having done this, it's the alternation generations that link the megaspore by way of that female gametophyte in the embryo sac, fertilization, and that microspore, that immature male gametophyte of pollen grain. So that's how it occurs. So with that, each of those spores do potentially give rise to that gametophyte. And of course, the cycle then continues. So in flowering plants, the diploid generation, that sporophyte, is larger and nutritionally independent of that haploid gametophyte generation, which is located within the flower and is microscopic and dependent upon that sporophyte generation. So keep that in mind. So up next, I'll get on down to the flowering plants, and here I'll begin with the way in which the flowering plants, how they flower. I'll go backwards just a bit. So the question is posed, how does a plant know that it's time to start forming flowers? So it's timing. So with that, it's a variety of environmental cues such as temperature and day length, and even with those that ensure proper timing, and different species are adapted to respond to distinct environmental cues. So the way it happens is via research done with a model called Arabidopsis thaliana. So when environmental conditions induce flowering, there are many genes that are activated or inactivated. And I'll leave it at that as far as this lecture is concerned, and now get to each part of the flower and its specific function. So this does piggyback off of what we've just done. So at the tip of the stalk is the receptacle on which some or all flower parts are born. So all 4 flower parts are important in the reproductive process, but only the stamens, which are the male organs, and carpels, which are the female organs, are direct participants in the sexual reproduction. Sepals and petals are sterile. So with that, the sepals, and this is all review from the 20th chapter class, so I'm going to pick up my pace here. Sepals, which make up that outermost layer, they cover and protect the flower parts when the flower is at developing bud. The sepals are leaf-like in shape, and they form and are often green in color. Some sepals, such as in the lily, are colored and resemble petals. So the collective term for all of the sepals is the term called calyx. So all sepals make up the calyx. Next inside in will be the petals. So the petals are broad, flat, and thin. So typical of which is for petals to be brightly colored, which are there to attract animal pollinators. Petals play an important role in ensuring that sexual reproduction will occur. Sometimes the petals fuse to form a tube or other floral shape, and the collective term for all petals is the corolla. So next we're going in is the stamen. Stamens are that male reproductive organ. Each stamen has a thin stalk called a filament, and at the top is the anther, that sac-like structure in which pollen grains are formed. For sexual reproduction to occur, pollen grains must be transferred from the anther to the female reproductive structure known as the carpel, and usually of another flower of the same species. More about that in just a moment. So at first, each pollen grain consists of 2 cells surrounded by a tough outer wall. One is that generative cell, and it divides mitotically to form 2 non-flagellate male gametes, known as sperm cells. The other is the tube cell, and it is what produces the pollen tube through which the sperm cells travel to reach the ovule. And that fourth and final world is, of course, the carpel. So the carpel is located in the center or at the top of most flowers. The carpels or bare ovules, which are those structures with the potential to develop into seeds, the mature ovule. The carpels of a flower may be separate or fused into a single structure. The female part of the flower, often called the pistil, may have a single carpel or a group of fused carpels. Each pistil has 3 sections. Going from top to bottom, it is that sticky stigma on which the pollen grain lands, the style, that net-like structure through which the pollen tube grows, and ultimately the ovary, the jug-like structure that contains one or more ovules that can develop into the fruit. So there is a great deal of variation in flowers. And I say a great deal of variation because the flowers are quite different. And there's a video that you should look at, not quite yet, but as we get there in the lecture, about the way in which the orchid wasp has co-evolved with its animal pollinator, with its, of course, plant, being the orchid. So with that, flower structure is important in plant identification. And of course, if flowers are present, flowering plant species are easy to evaluate in the field. Hence is when you have the plant collection, it is stated that you must have specimens collected that have reproductive structures, because it is those reproductive structures that are used for us, for you, for anyone on Earth, to identify plants to species. If you have any questions, please let me know. I'll now continue on with that female embryo sac. So female gametophytes, which can also be called that embryo sac, are produced in the ovary. The male gametophytes in the anther. So with this, just make sure you review what was discussed in the 27th and 20th chapter. I'm saying that to say this. Each young ovule within an ovary contains a diploid cell called that megasporocyte, which undergoes meiosis to produce 4 haploid megaspores. 3 of them will disintegrate. You all should remember this from meiosis, such as a polar body would have been. The 4th is that functional megaspore and divides mitotically to produce that multicellular embryo sac, which is that female gametophyte. So with that, within the embryo sac, which is embedded in the ovule, it typically contains 7 cells with 8 haploid nuclei. 6 of these cells, including the egg cell, the female gamete, and they contain a single nucleus each. And then there's a large central cell that has 2 nuclei called polynuclei, which is why I mentioned polar nuclei as I described the process of double fertilization. The egg and both polynuclei participate directly in fertilization. Pollen sacs within the anther contain numerous diploid cells called microsporocytes, each of which undergoes meiosis to produce 4 haploid cells called microspores. Each microspore divides mitotically to produce an immature male gametophyte, also called a pollen grain. That pollen grain consists of 2 cells, as mentioned just moments ago, the tube cell and the generative cell. The pollen grain then becomes mature when the generative cell divides to form 2 non-motile. I repeat, 2 non-motile sperm cells. And I say non-motile because there is no longer a need for water for fertilization for those sperm cells that were motile to swim, in the case of course, the bryophytes and the seedless vascular plants. And this figure shows everything I've just done. If I were you, I would make sure I review this figure to ensure that you understood what was just given to you. And this is one of those key points found on page 785. I'll now continue. So we'll now get to what is called pollination. I'll make pollination blunt. Before fertilization can occur, pollen grains must travel from the anther, where they form, to the stigma, the sticky stigma as I sometimes say. The transfer of pollen grains from anther to stigma is known as pollination. Plants are self-pollinated if pollination occurs within the same flower or a different flower on the same plant. when pollen grains are transferred to a flower of another individual of the same species. This is termed cross-pollinated. Flowering plants accomplish pollination in a variety of ways. And you say, okay, so how does it occur? Pollination occurs via the following: beetles, bees, flies, butterflies, moths, wasps, and other insects pollinate many flowers, and even animals such as birds, bats, snails, and there are included here small non-flying mammals. Yes, there are mammals that do not fly, which are small, that are also animal pollinators, such as marsupials, primates, and rodents. And then ultimately, wind is an agent of pollination for certain flowers, and water in a few aquatic flowers. So many plants have evolved mechanisms that prevent self-pollination. In plant sexual reproduction, the two gametes that unite to form a zygote may be from the same parent plant or from two different plants. The combination of gametes from two different plants increases the variation in offspring, and this variation may confer a selective advantage. Some offspring, for example, may be able to survive environmental changes better than either parent plant can. So evolution has resulted in a variety of mechanisms that prevent self-pollination and thus prevent inbreeding, which is the mating of genetically similar individuals. Inbreeding can increase the concentration of harmful genes in offspring. Some plants, such as asparagus and willow, have separate male and female individuals. So the male plant has those staminate flowers that lack carpels, and the female plants have the pistillate flowers that lack stamens. Other species, and before I get to the other species, that's termed being dioecious, having separate male and female plants. Other species have flowers with both stamens and pistils. However, the pollen is shed from a given flower either before or after the time when the stigma of that flower is receptive to the pollen. Hence, these two mechanisms promote outbreeding or outcrossing, which is the mating of dissimilar individuals. So with that, just keep in mind that even plants, though they aren't able to think, have mechanisms, have ways of promoting outbreeding and preventing inbreeding. So I won't do anything with self-incompatibility at the moment. What I will now get to are animal pollinators. So animal pollinators and their plants they pollinate have had such close interdependent relationships over time, they have affected the evolution of certain physical and behavioral features in one another. What I'm getting to here is the process of coevolution. So coevolution describes such reciprocal adaptation in which two species interact so closely they become increasingly adapted to each other as they each undergo evolutionary change by means of natural selection. So let's examine some of those now. Flowers pollinated by animals have various features to attract their pollinators, including having showy petals that visual attractant and scent, an olfactory attractant. One reward for the animal pollinator is food. So pollen grains are a protein-rich food for many animals. As they move from flower to flower, searching for food, pollinators inadvertently carry along pollen grains on their body parts, helping helping the plants class reproduce sexually. Some flowers produce nectar, a sugary solution, in special floral glands called nectaries. Pollinators use nectar, which is rich in amino acids, alkaloids, and other floral chemicals, as an energy-rich food. Some nectars contain alkaloids such as nicotine, to prevent animal pollinators from drinking all the nectar in a single visit. Nicotine is poisonous to all. Thus, pollinators are encouraged to visit multiple flowers, and each flower has multiple pollinator visitors. Again, each flower has multiple pollinator visitors. So it's been estimated by biologists that insects pollinate about 70% of all flowering plant species. Going back to the estimated 300,000 species of plants, to estimate 70% of that, please write that down on paper right now. That is a lot of plants, you know. Bees are particularly important as pollinators of crop plants. Crop pollinated bees provide about 30% of human food. In fact, I was on a trip to New Orleans one spring break. It wasn't this year. However, as I drove back from New Orleans on Interstate 10, I just happened to look to my right because I was passing a truck, an 18-wheeler. And I don't know if you all have heard, but apparently almonds flower and fruit. The tree does. The almond tree. Well, to ensure that the almond tree is appropriately pollinated, bees from all over the country are shipped to California. So just by chance, I happened to look at that truck and I saw boxes. Not just cardboard boxes. And it wasn't cardboard boxes at all. I saw boxes of bees on Interstate 10 heading east. It blowed my mind to see such things. So if you did not think bees were important, they are some of the most important animals on Earth because, as I just mentioned, they provide us with at least 30% of our food. Plants pollinated by insects often have blue or yellow petals. So with that being stated, the insect eye does not see color the same way the human eye does. Most insects see well in the violet, blue, and yellow range of visible light, but do not perceive red as a distinct color. So, flowers pollinated by insects are usually not red. Insects also see in the ultraviolet range. So please see the figure being shown now. So ultraviolet markings on this Eden primrose, they draw the attention to the center of the flower where insects find nectar and pollen grains. So we just look at a flower such as this and only see yellow. So if you look at it under the ultraviolet radiation, this provides the clues about how the insect eye perceives it. So the light blue portions of the petals may appear to the purple to a bee, whereas the dark blue inner parts may appear yellow. How amazing. So as I was stating there, insects also see the ultraviolet range, wavelengths that are invisible to the human eye. So insects see ultraviolet radiation as a color called bee's purple. Many flowers have dramatic UV markings, such as what you see here, that may or may not be visible to humans, but that direct insects to the center of the flower where pollen grains and nectar are. So insects, they have a well-developed sense of smell, and many insect-pollinated flowers have a strong scent that may be pleasant or even foul to humans. For instance, the carrion plant. It's pollinated by flies and smells like rotting flesh in which flies lay their eggs. As flies move from one reeking flower to another looking for a place to lay eggs, they transfer pollen grains. Birds, such as hummingbirds. If you think back, yes. So mentioning the hummingbirds, they are important animal pollinators too. The flowers pollinated by birds are usually red, orange, or yellow because birds see well in this range of visible light. Because most birds do not have strong sense of smell, the bird-pollinated flowers usually lack a scent. On to the next. Bats. They feed at night and do not see well, and they are important pollinators, and this is particularly in the tropics. Bat-pollinated flowers bloom at night and have dull white petals and a strong nighttime output of scent that usually smells like fermented fruit. Nectar-feeding bats are attracted to flowers by the scent. They lap up the nectar with their long, extendable tongues. As they move from flower to flower, they transfer pollen grains. An unusual adaptation that encourages pollination by bats has evolved in the tropical vine Mucuna holtani. When the pollen in a given flower is mature, the concave petal lifts up, and the petal bounces from the bat's echolocating calls back to the bat, helping it locate the flower. How amazing. So specialized features such as petals, scent, and nectar attract pollinators that have co-evolved with adaptations of animal pollinators. Specialized body parts and behaviors adapted animals to aid pollination as they obtain nectar and pollen grains as a reward. For example, coevolution has selected for bumblebees' hairy bodies, which catch and hold sticky pollen grains for the transport from one flower to another. Coevolution may have led to the long, curved beaks of the iwi, one of the Hawaiian honeycreepers, that inserts its beak into the tubular flowers to obtain nectar. The long tubular corolla of the flowers that the iwis visit probably came about through coevolution. During the 20th century, some of the tubular flower species became rare, and is largely as a result of grazing by non-native cows and feral goats, and about 25% of those species, the lobelia species, have become extinct. So the iwi now feeds largely on the flowers of the ohawi tree, which lacks petals, and the ohawi bill appears to be adapting to this change in feeding preference. So the comparison of the iwi bills in museum specimens collected in 1902 with the bills of the live birds captured in the 1990s show that the bills were about 3% shorter in the 1990s than in 1902. Plants and the behavior of their animal pollinators have co-evolved, sometimes in bizarre, complex ways. The flowers of certain orchids resemble female bees in coloring and shape. The unpollinated flowers secrete a scent similar to that produced by female bees. The males are irresistibly attracted to it. The resemblance of the orphis flowers and female bees is so strong that male bees mount the flowers and try to copulate with them. During this pseudo-copulation, a pollen sac usually attaches to the back of the bee. When the frustrated bee departs and tries to copulate with another orchid flower, pollen grains are transferred to that flower. Interestingly, once a flower has been pollinated, it emits a scent like that released by female bees that have already been mated. Male bees have no interest in visiting flowers that have been pollinated, just as they would lose interest in female bees that have already been inseminated. So there you have it. So please see the video at Canvas that shows you what you're seeing here at part F of the orphis. So it's an amazing video, and the video is only an approximate 2.5, I think it's 2.5 minutes long. As you look at A, of course, those are honeybees, or at least a honeybee, Apis mellifera, pollinating a Scotch broom flower. At B, you're seeing a tiger swallowtail butterfly, and in this case, it's obtaining nectar from butterfly weed flowers called Asclepias tuberosa. And if you can, I say please plant more milkweeds, particularly the native milkweeds, to ensure that the monarch butterfly can, of course, persist. At C, what you're seeing there is an Australian long-nosed lacet beetle. obtaining nectar from, of course, the Australian tea tree. So, having mentioned all of this, I'll say that table 37.1 should be on your test. And what you'll do is go through each of those. You go from animal pollinator to the common flower, color to the scent, the pollinator reward, and the time of flowering. So you'll fill in those. I won't tell you which ones to fill in, but I'll just say, hey, learn it. That way you know that the bee is, of course, out during the day, looking for nectar and or pollen, looking for a floral scent, and typifying the flower that is yellow, blue, or purple. So just make sure you take your time. You can find this in your textbook on page 788. Alright, up next, self-flowering plants depend on wind to disperse pollen. So as I say such, I'll say flowering plants such as maples, ragweed, grasses, and oaks are pollinated by wind. So those wind-pollinated plants produced many small, inconspicuous flowers. They have no large, colorful petals with scent or nectar. Some have large, feathery stigmas, and presumably it's there to trap that wind-borne pollen grain. Because wind pollination is a hit-or-miss affair, the likelihood of a particular pollen grain landing on the stigma of the same species of flower is slim. Hence, those wind-pollinated plants produce large quantities of pollen grains, copious quantities of pollen grains, which increases the likelihood that some pollen grains will land on the appropriate stigma. So if you look closely here, you're seeing, of course, those catechins, those catechins that are providing the pollen. And of course, this is like what has you sneezing sometimes when you're outdoors. So with this is from an oak tree. And of course, it blows and blows and blows. So up next, we'll get to fertilization with seed and fruit development. So as I mentioned it, after the pollen grains have been transferred from anther to stigma, the tube cell, one of the two cells in the pollen grain, grows into that thin pollen tube down through the style and into the ovule in the ovary. So the question might be asked, how does the pollen tube know where to grow? So it's by molecular signaling, and this happens by way of the synergid cells located adjacent to the egg that guide the growing pollen tube toward the ovule. So there are two attracting signals called lures, L-U-R-E-S, and they're both small polypeptides that have been studied in wishbone flowers, and that there are ongoing investigations to detect others and understand the mechanism of action. So let's get down to this unique double fertilization process in flowering plants. On your test, prepare yourselves to, of course, describe the process. Let's continue. So as double fertilization occurs, class, please bear in mind that it's the egg within the ovule that unites with one of the sperm cells. This forms a zygote, that fertilized egg, and that develops into the embryonic plant in the future seed. I'm going to pause for just a moment, class. On your test, you won't just describe this process. You will also provide the importance of what is created by way of this process. I'll begin again. The unique double fertilization process that occurs in flowering plants happens by way of that egg within the ovule, which unites with one of the sperm cells. That forms a zygote, otherwise known as a fertilized egg. It will then develop into that embryonic plant contained within the future seed. Please write this down. The two polar nuclei in the central cell of the ovule fuse with the second sperm cell to form the first cell of the triploid, that's 3 indole, endosperm. The endosperm is the tissue with nutritive and hormonal functions that surround the developing embryonic plant in the seed. This process in which two separate cell fusions occur is called double fertilization. This process is called double fertilization. So it is, with a few exceptions, unique to the flowering plants. So a type of fertilization has been reported in two gymnosperms, being ephedra and nectum. So after double fertilization, the ovule develops into a seed, and the surrounding ovary develops into a fruit. Please, class, do not forget that. So embryonic seeds. So the embryonic development in seeds is orderly and predictable. The following plants package a young embryonic plant, a young embryo, complete with stored nutrients in a compact seed, which develops from the ovule after fertilization. The nutrients in seeds are not only used by germinating plant embryos, but also eaten by animals, including us, called humans. So development of the embryo and the endosperm following fertilization is possible because of the constant flow of nutrients into the developing seed from the parent plant. Its cell divisions of the fertilized egg to form the multicellular embryo proceed in several ways in flowering plants. So let's get to dicot embryonic development. So there are two cells, and I'll leave it at this. There are two cells, the basal cell and that apical cell, that are formed as a result of the first division of the fertilized egg. And this is what establishes polarity, meaning the direction in the embryo. The large basal cell located toward the outside of the ovule typically develops into the A suspensor, and that's that embryonic tissue that anchors the developing embryo and aids in nutrient uptake class from the endosperm. The apical cell, which is toward the inside of the ovule, develops into the plant embryo. So it's still under research about what molecular differences between the apical cell and the basal cell determine the initial molecular signals that are involved in establishing polarity. So initially, the apical cell divides to form a small cluster called proembryo. So those proembryo cells happen by way as that cell division continues, and a sphere of cells, often called the globular embryo. So the cells begin to develop into specialized tissues during this stage, when the dicot embryo starts to develop its two cotyledons, or its two seed leaves. So it has two lobes, and it resembles a heart, the heart stage. And then during the torpedo stage, the embryo continues to grow as those two cotyledons elongate. So as the embryo enlarges, it often curves back on itself, and then it crushes the suspensor. So having gotten this far in the lecture, I think I'm going to pause the lecture here. Then I'll pick up and finish this lecture for next class, next time. To help you all out as far as your studies go, please, class, make sure you have prepared yourselves for the following exam by studying the following figures from Chapter 34. There you are, Figure 34.4. By way of Section 34.1, Section 34.3, 34.4, and 34.5. From Chapter 35. From Chapter 35, make sure you all have known and learned those figures called Figure 35.1, Figure 35.2, and Figure 35.5. And those, of course, are done by way of Section 35.1 and Section 35.2. And then from Chapter 36, make sure you all have learned Figure 36.3 and 36.8 by way of Section 36.1 and 36.2. And all of that should be done along with learning Figure, I think it's 28.13 in the 20th chapter, which is that life cycle for angiosperms. So this has been your instructor, Skylar Huff, and this has been the first part of the 37th chapter. Thank you all again. Prepare well for your test.